embryonic development in human being. The human embryology is a study of human development during the first 8 weeks after fertilization. Human embryology is the study of human development during first 8 weeks after fertilization. From the beginning of the ninth week, the developing young one is called fetus. From ninth week onwards, the developing young one is said to be fetus. So that type of cleavage here, it is called holoblastic type of cleavage. Then because microlecital condition of the egg, let's start the egg, let me draw the egg to you. The secondary oocyte and after meiosis 2, that ovum is formed and that was fertilized with the nucleus, male pronucleus, and zygote is formed. That condition we discussed in the fertilization topic. And now the situation is like this, that a little perivitelline space and zona pellucida is present. So this is a layer, zona pellucida is still there. Zona pellucida after fertilization and after the formation of egg, still zona pellucida is there, and the little amount of yolk substance is present here. The little amount of yolk substance, so we call microlecital egg. Such kind of egg is said to be microlecital egg. So, due to the presence of very little amount of yolk substance is very little. So that's why the total zygote is going to participate in the division. That during the division process, the whole zygote will participate in the division. We call it as holoblastic cleavage. Holoblastic cleavage due to this microlecital condition. Holoblastic cleavage is due to microlecital condition. And that the cleavage, being it belongs to the human, belongs to the deuterostomia, radial indeterminate type of cleavage is seen over. The cleavage patterns to be remembered: holoblastic, radial, and indeterminate type of cleavage. And after the division, after the cleavage, the whatever the blastomeres are formed, the cells are formed which are equal in size. So that's why we say this as equal cleavage: holoblastic, radial, indeterminate, equal cleavage with microlecital egg. With we need to remember it as microlecital egg. Okay, so this is the condition. This is about the cleavage. Okay, so then let's move on to the next one. So then the divisions will go on: 2 cell, 4 cell, 8 cell, 16. That 16 divides and form the 32. At this time, that the number of daughter cells when reaches to the 16 to 32, the solid ball of cells, now it is called MORULA. Now it is said to be MORULA. With 16 to 32 cell stage, it is said to be MORULA. Okay, so still our said zona pellucida is there still. Zona pellucida is there. So then this, what do you say, the solid ball of cells is called MORULA. This is MORULA. Okay. Then it look like mulberry. The MORULA look like mulberry. At this stage, the cells start to bind firmly together, and this process is called compaction. We'll draw the compaction here. Compact. What is compaction? The cells are becoming compact. We say compaction. Okay, so let me draw the compaction condition here. That cells become bound tightly together, and the outer surface of the blastomere flatten against each other. So let me show you. Here it is. Outer surface of the cells are becoming flatten each other, and they bind each other with the tight junctions. They bind each other with the tight junctions. This tightly packed arrangement is stabilized. This tightly packed arrangement is stabilized by tight junctions that form between the outer cells of the morula. So now it is shown to you, tight junction. This is tight junctions are getting formed. Okay. Next, the cells within the sphere. The cells within the sphere are going to loosely arrange. We say they form the gap junctions. The cells within the sphere is going to form connected each other that with a gap junction which helps in the better passage of substances in between them. The better passage of substances in between them. The tiny embryo is still surrounded by glycoprotein layer. The tiny embryo is still surrounded by glycoproteinous layer of the egg, Juna pellucida. Okay, so that is still surrounded by zona pellucida. At the completion of cleavage or blastulation, the embryo has a central cavity called blastocyst. You can't see little bit, very small amount that cavity will find that slowly it is going to be much organized. That means the blastocyst is going to be expand over. Now I can show you the blastocyst. Okay, so that at the completion of cleavage, blastulation, the embryo has a central cavity called blastocyst. Now the embryo has a superficial flat cell layer and the superficial flat cell layer, this red color, and inner cell mass, the blue one, this inner side, but what do you say? That inner cell mass. The cells on the exterior of this early embryo develops into trophoblast. This is trophoblast and this is inner cell mass. Okay, so let me label this first for you, the trophoblast and inner cell mass. So, so this is, here it is, I am trying to label trophoblast. Okay, so this is trophoblast. So, and this is inner cell mass. Trophoblast and inner cell mass. Okay, so that the thing I just made two diagrams to be here. This is the morula, and now I shown here how a morula will turn into the stage called blastocyst. How the morula turned into the stage called blastocyst. I just shown to you. So let me read you once again. For this, that means I am trying to show the visuals of the each and every text sentence given here. Then when the number of daughter cells is 16 to 32, the solid ball of cells is called morula. This is the morula. Okay, so this is the morula. Okay, so then it looks like mulberry. It looks like mulberry. At this stage, the cells start binding firmly together, and this process we say compaction. That means you see the evolution of the cells from here to here. That compaction is happening. The tightly packed arrangement, the compaction, the cells of the morula become bound tightly together, and the outer surface of the blastomere flatten against each other. This tightly packed arrangement is stabilized by tight junction. Tightly packed arrangement is stabilized by tight junction that form between the outer cells of the morula. The cells within the sphere form gap junction. Here in this area, I am showing this, in this area, gap junctions are formed, which help in the better passage of substances between them. The tiny embryo is still surrounded by the glycoproteinous layer of the egg, zona pellucida. This is the zona pellucida. At the completion of the cleavage, the blastulation, the embryo has a central cavity that we say the blastocyst. Now the embryo has a superficial flat cell layer and inner cell mass. Superficial flat cell layer we call trophoblast, and inner is said to be formative cells or inner cell mass. Here I just shown here. Whatever shown with red color, that is trophoblast, and inner cell mass. That means we'll take one more fresh diagram here to understand much better. That so let me remove Then, so this is zona pellucida. This is zona pellucida. Then below that, the tightly packed cells. That means the flattened cells which are tightly packed each other. What do you say that as? Trophoblast cell. And inner loosely aggregated cells. The inner loosely aggregated cells we say inner cell mass. Okay, so this is zona pellucida and this is trophoblast and these are inner cell mass. These are the participants in this session, in this paragraph. Okay. The cells of the exterior of this early embryo develops into trophoblast or trophectoderm, this one. Okay, then outer epithelium of the blastocyst. Then inner cell mass, this is the green one. This is inner cell mass. Okay, so zona pellucida is as usual. So that is about the blastocyst is formed while the early embryo passes through the fallopian tube. That means you people need to know the passage also. We learned that fertilization process occurs in fallopian tube, isn't it? The fertilization occurs in the fallopian tube. Okay, then zygote is dividing now 2 cell, 4, 8, 16, 32. In the mean period of time, it is also passing from the fallopian tube towards the uterus. It is passing towards the uterus. In the mean period of time, this development is going on. Okay, so that's it. The development is going on, still going on. Then the next, let's continue. Let's continue. So this is all about the morula and how the morula is turned towards the blastocyst. Now let's start. Then whenever that, okay, let me show you once that particular procedure that we learned in the fallopian tube. This is the fallopian tube and now we are moving to the uterus. The uterus is with three layers, that epimetrium, myometrium and innermost layer of the uterus is called endometrium. And now the blastocyst should come here for what? For implantation process. That means it is need to make the connection with the mother, isn't it? So now where all our said events are happening, here all other said events are happening. Zygote, 2 cell, the 4 cell, 8 morula is formed. Then the morula is now turned into the blastocyst. All that events are happening here. That means when the development is going on, from the fallopian tube to the uterus, it is moving. Okay, so now the some fluid secreted into this cavity. So when the some fluid will be secreted into this cavity, that whenever the blastocyst reaches here into blastocyst, the stage we call morula turned into blastocyst now, that with trophoblast and inner cell mass, that whenever it reaches into the blastocyst, that blastocyst reaches into the uterine cavity, the some fluid secreted into this cavity, then the embryo acquires the shape of the cyst. Now we can say the fully as blastocyst. Okay, so in the blastocyst. Okay, so now let me show you the blastocyst clearly. So, but I should draw zona pellucida because still zona pellucida is there. Okay, so now when it is dropped into the uterine cavity, see that some confusion will go on continuously. So that's why just focus here. So now this is the condition. Okay, so this is zona pellucida and this is trophoblast, inner cell mass. What difference we found with the previous one? This cavity, blastocele cavity is much extended because that the fluid of the uterine cavity, when it is started entering into the uterine cavity, the fluid of uterine cavity is accumulated over. Now it appears like cyst. We call it as blastocyst. The process of formation of space around the formative cell. This formation of space around the formative cell is called cavitation. This process of formation of space around the formative cells is called cavitation. The inner cell mass, this is the inner cell mass. Let me show you. This is the inner cell mass, which is even called formative cells. Inner cell mass or formative cells, which give rise to the embryo proper. which give rise to the embryo proper. That means these kind of words will be understood when we reach to the end. That means some fetal membranes, the embryo proper and some fetal membrane. So talking about the future of the inner cell mass. So what happens with this inner cell mass? This inner cell mass is going to form the embryo proper. That means total embryo. Then some fetal membrane. That from here itself, some fetal membranes are also going to be formed. So that you need to understand in that manner. So when we go future, that we'll find them. Then so these cells constitute the embryo blast. So because of it is inner cell mass form the embryo proper and also some fetal membranes. So these cells constitute embryo blast. They are the source of all the pluripotent embryonic stem cells. They are the source of all the pluripotent embryonic stem cell lines. In addition to the embryo proper, the inner cell mass gives rise to extra embryonic membranes also. That means multiple events in one place it is given. So first of all, let us know what is pluripotency. So behind the pluripotency, we have one more word, that is totipotency. Totipotency. What is totipotency? Forming all types of cells, we say totipotency. That means zygote. Zygote form everything. Zygote form embryo, zygote form fetal membranes. Likewise, that we say totipotency. Now the inner cell mass is having the capability to produce some fetal membranes and also the embryo blast. That also embryo blast. So that's what I mean. Also, it formed the embryo, a future embryo, and extra fetal membranes that also it can form. So that's why what do you say? That is called pluripotency. Okay, we'll discuss separately the totipotency, pluripotency, multipotency, unipotency. That all we'll discuss separately. But you just try to understand the series and sequence you need to follow over. The only sequence you need to follow over. So what we learned till the time? We learned till the time zygote divides into two cells. That kind of cleavage is said to be holoblastic, equal, radial, indeterminate cleavage. When it all dividing, that leads to the formation of MORULA. That still zona pellucida is there surround the MORULA, and in the MORULA, the cells will undergo the compaction process. The compaction occurs, and the results outer layer of the cells will tightly pack each other with tight junctions, and inner loosely aggregated cells will form the gap junction with a small cavity the leaf, and still that they surrounded by a layer called zona pellucida. As usual, zona pellucida is there still, and at this condition, from the fallopian tube, it is now dropping into the uterine cavity. A small cavity, a small crevice, that what is a blastocyst, which is appeared into that, the fluid is moving on. When the fluid moves on, so what happens? A blastocyst is appeared. A blastocyst. That means the trophectoderm or trophoblast is as usual inner cell mass, which is pushed at one side, and this is the blastocyst. So at this stage, we call it as blastocyst. It is said to be a blastocyst. Okay. So remaining these kind of issues, you people will get confusion here. That issues we have to discuss when we move forward. We have to come back again. We need to see here because they are saying about the future. We don't know the future till the time. So that's why let us wait. So events are like this. Okay. So till the time these are the events are happening. Okay, next, let's continue. Let's continue. Implantation process. Implantation process. Okay, so for that, I'm going to draw the uterus. Uterus with three layers. What are the three layers? Epimetrium, myometrium, and that endometrium layer. Epimetrium, myometrium, then that endometrium layer, thickened endometrium layer with blood vessels and blood capillaries. So this is the condition. Okay, this is the condition. Okay, so now let us take our blastocyst also. That I'll draw the blastocyst for you. That it is surrounded by zona pellucida till the time, and we know the trophoblast, and that inner cell mass is also there. This is the blastocyst stage with blastocyst, So now it has to come for implantation here. Okay, now it is dropped into the uterus for implantation. So for that, what needs to happen? Let us see. The blastocyst hatches out. That means the blue one inside. This is blastocyst hatches out of the envelope, the zona pellucida, by lysing its wall with the help of an enzyme called stripsin. With the help of an enzyme called stripsin, this that zona pellucida, the red one, is now moving out. You see here, the zona pellucida. When stripsin, enzyme stripsin makes them to get disappear. So that need to be disappear for what? For implantation. So with the help of enzyme stripsin, the zona pellucida is getting disappear. Later, the zona pellucida gradually disappear. The cells of the trophoblast stick to the uterine endometrium. Now, that exposed one, who is that? Who is exposed now? Zona pellucida is gone. So who is exposed now? Trophoblast is exposed now. So this trophoblast is moving on. So let me draw the inner cell mass also, not to get confused. These trophoblast cells, the cells of the trophoblast stick to the uterine endometrium. Now they are moving. They stick to the uterine endometrium and form a part of the fetal part of the placenta. Fetal part of the placenta. So that we'll discuss, fetal part of the placenta. So then, simply what I can say at present here, that the trophoblast layers are dividing rapidly and reaches into the endometrium layers of the mother. They reaches into the endometrium layers of the mother. Okay, next, the trophoblast invades the endometrium. The trophoblast invades the endometrium of the uterus. In human, the process of implantation begins on the sixth day after fertilization. Sixth day after fertilization. This process of implantation is aided by, that means penetrating into this also requires some enzymes, aided by proteolytic enzymes produced by the cells of the trophoblast. The trophoblast thickens through cell division and the wall of the trophoblast develop trophoblastic villi. These are the trophoblastic villi. Develops trophoblastic villi that branch and project into the highly vascular uterine endometrium. They are projecting into the highly vascular uterine endometrium. For what? To draw the nourishment for the embryo. The trophoblast differentiate into inner cell layer called cytotrophoblast. Let me show you this area in microscope. The trophoblast layer, that it is differentiated into inner cytotrophoblast. That means the cell layer, like the cell membranes are present. This is cytotrophoblast, inner cytotrophoblast, and outer syncytiotrophoblast. This is called, which is penetrating into the endometrium layer, is called syncytiotrophoblast. We can say. So let me read. So that cytotrophoblast and inner, then cytotrophoblast and syncytiotrophoblast. Okay. So let me show you the diagram. This cytotrophoblast, the cellular trophoblast, which is also called layer of Langhans, That cytotrophoblast is also called layer of Langhans, consisting of cuboidal epithelial cell. This one we are talking about. The layer of Langhans and consisting of cuboidal epithelial cell. And the outer syncytiotrophoblast, a layer of fused cells. You can see here the cell membranes are not present. Cell membranes are absent. Then the embryo along with its membranes, now it is called conceptors. Now the embryo along with all these membranes is considered as conceptors. The total process we call implantation. Okay, so let me say once again, the blue color, the blue, you need to recognize it. This is trophoblast. What it is doing? It undergoes continuous divisions mitotically, and the mitotic divisions makes them that two types of trophoblast layer like syncytial trophoblast and cytotrophoblast. The syncytial trophoblast penetrating into the endometrium layers of the mother, then for the to draw the nourishment. In future times, this is only we are going to say it as placenta, a connection, a part of placenta. Okay. So that straight plain talk we are doing actually. Now, that just following the embryo, the formation of bilaminar embryonic disc. The formation of bilaminar embryonic disc. Okay, implantation is over. That implantation is over. Here we need to include some decidua information. That what we discussed yesterday. The decidua information. So I'll make space myself. That for decidua information. So after the implantation, that I am making some space here to show after the implantation how the full scenario can be seen over. Making space myself. Let me say once again, this is epimetrium, and below that myometrium is present. That myometrium, that uterus with three layers, epimetrium, myometrium, and innermost layer can be called endometrium. After the completion of implantation, so what is the situation that we need to see here? This is the uterine endometrium layer, the two-dimensional diagram which we are drawing. So now this is the embryo, our so-called blastocyst. Okay. So this blastocyst which contain the trophoblast and inner cell mass. So as we said, the trophoblast is divides and penetrates into the mother, which forms syncytial trophoblast to penetrate inside. Okay, so this deeper penetration, finally what happens here, the deeper penetration of these, that endometrium layer completely surrounds the embryo, which means embryo completely penetrated and enters into the uterine wall. You should understand like that. Okay. So in this case, we call this layer as decidua. This is called as decidua. Okay. So this is said to be decidua. So I should increase the size decidua for that. Okay, so that who is decidua, whom we call decidua? Endometrium layer, the red color, the total red color we say decidua. That means endometrium layer. Endometrium after the implantation is considered as decidua. Then in this, that decidua here, decidua here, and decidua. Little need to show here. This is decidua here. That we got some names yesterday, isn't it? So what is this? Decidua basalis. Decidua basalis. Okay. So what is decidua basalis? Location we need to say here. Let me write the total. Decidua capsularis. Decidua capsularis. And this is called decidua parietalis. Decidua basalis, decidua capsularis, decidua parietalis. So location, we know this green one. What is that? Myometrium. And we know this is embryo. So we need to give the address of the decidua basalis. It is present between the myometrium and embryo. So that endometrium layer present between the myometrium and embryo is called myometrium and embryo. Let me use other color to show this. Myometrium and embryo. So what do you say it as? Decidua basalis. Then we know this is the lumen of uterus. Okay. So this is embryo, this is lumen. So here it is decidua capsularis. So address we are giving decidua capsularis between the embryo and the lumen of uterus is decidua capsularis. Decidua parietalis, so simple. Other than the site of implantation, this is the site of implantation. Other than the site of implantation, wherever you go, that place we say decidua parietalis. So need to be remembered that After the implantation, endometrium layer itself called decidua. Then the decidua, endometrium layer present between the myometrium and embryo is called decidua basalis. Endometrium layer between the embryo and the lumen of uterus is called decidua capsularis. Other than the site of implantation, that endometrium layer is called as decidua parietalis. So this is the total story that we need to understand. Okay, so that is decidua. Little I am adding actually here. That little I am adding. So now let's move back to our story. That we said embryon, the implantation is over. After the implantation, endometrium layers became decidua that I shown, I explained decidua basalis, capsularis and parietalis. Now we'll discuss about the blastocyst after the implantation, what it is doing. Implantation of the blastocyst is completed by the end of second week. The inner cell mass forms into a disc called embryonic disc that we can't show in the diagram. That means what I say, that if you show me a ball, that if I show you a disc, both will be drawn in the same manner, isn't it? So that the same diagram I have to draw. That means that is disc like. That inner cell mass forms into a disc called embryonic disc or germinal disc. The embryonic disc, as usual, has an outer group of cells called epiblast. So, first of all, let me show you the trophoblast first of all. Then, after, we need to go further. Epiblast and hypoblast cells. Epiblast and hypoblast cells. Okay, so epiblast and hypoblast. So this is epiblast. The blue one is the epiblast. Okay, so then the green one, as I said to you yesterday, that there is some confusion, two-dimension or three-dimension diagrammatic problem. That means you need to understand that their positions, epi, up. That means that, as I said to you, if there is an object above on this, we'll have one more object that like that you need to imagine. This is epiblast and this is hypoblast. That three-dimensional angle you need to think over to understand this kind of labeling that I shown to you earlier. Okay, so the embryonic disc has an outer group of cells called epiblast, outer group, blue one, epiblast, and that in future it is going to form the ectoderm, and inner layer of the cells called hypoblast. Okay, the primitive endoderm made of cuboidal cells. The hypoblast lines surface facing the blastocyst cavity. That means these hypoblast cells slowly will come here. Okay. So first of all, let me explain that what is given here. The hypoblast lines surface facing the blastocyst cavity. So I'm showing with arrow. Hypoblast lines surface facing the cavity. They are facing towards the cavity. It is the future extra-embryonic endoderm. So future, okay, we'll discuss that. Then hypoblast is pushed down. Now what do you say? Delamination. Blue separates from the green. Blue means epiblast. Green means hypoblast. Delamination. We know lamination means attachment. Delamination means here the cells of the hypoblast will separate from the epiblast and reaches into the cavity, and they lines the trophoblast or trophectoderm. That what do you say? It has delamination. That hypoblast is pushed down and it forms the lining of the yolk sac. Okay, so that I'll show you the full diagram again. So I'll draw you the bigger diagram. So this is the trophoblast. No way, no theoretical. The trophoblast is as well. Now we are in the complete paragraph. We are discussing about epiblast and hypoblast only. So this is the hypoblast and inner. Now I changed the color, isn't it? Okay, so let me use the same color. So that what I have to draw hypoblast. Okay, leave it. Let us draw the fresh diagram. Yeah. So I need to draw the hypoblast like this. This is hypoblast. And inner, who is the inner one? That is epiblast. So this is epiblast. Okay, the blue one. So now what is saying? The hypoblast cells, the hypoblast cells will go for delamination, separation. Then what they do? They're going to enter into the cavity which the lines below the profectoderm, the lines below the profectoderm. So the so-called yolk sac, they are going to form here. That let me read. The hypoblast push it down. That I shown you how they push it down. Delamination. It formed the lining of the yolk sac. Now this is nothing but yolk sac. We don't have any importance for yolk sac in case of human being because we are drawing nourishment from the mother, isn't it? Uterine endometrium layer. Already we are seeing the implantation. So that yolk sac doesn't have any importance. If you say birds, birds have the having more importance to the yolk sac. That means they provide nourishment to the developing embryo. The story is different. Okay, so now we are discussing about human being. Now the embryonic the disc with epiblast and hypoblast, it is considered as bilaminar embryonic disc. It is considered as bilaminar embryonic disc. Okay, what next? The further development involves the gastrulation process. The further development we have to go with gastrulation process. Okay, now let me move towards the gastrulation. Here you need to be little bit careful here for gastrulation. Gastrulation is an important dynamic process in the development of the early embryo, which involves movement of cell masses to their definitive positions in the embryo and form their three primary germinal layers. These movements are called morphogenetic moments. Let me say once again. It's a dynamic process, development of early embryo, which involves the movement of cell masses to their definitive position, movement of their into definitive positions in the embryo and form three primary germinal layers. Okay, so for that, we need to go for how does the thing happen. So I think you remember the epiblast layer. So epiblast we drawn with blue color, isn't it? So let me draw the same color. Let me follow the same. Epiblast. Here I have to add something. The epiblast, till the time it is discoblastula, isn't it? Now, this discoblastula is turning towards the development on one side that makes the oval shape and which it makes a primitive knot and a primitive streak, primitive groove, that primitive pit that I said to you earlier in the yesterday's class that on cake, if you with the knife, if you make a hole, the hole I can say primitive pit. Okay, so the border of the hole we can say primitive knot. Then this line, then after hole, what you have said to you do that the with the cake you do make a line like this in the cake itself. So that what you can say primitive groove. That means you should imagine here. You should imagine the primitive streak, primitive knot, primitive groove, primitive pit, that primitive folds. That all the things you need to imagine. Okay, so that let me draw the primitive streak knot. Okay, so then what is this exactly? The blue one. You need to understand that is epiblast. That what I made till the time. This is epiblast. Okay. So actually there are other layers are also present, but for our comfort we are not drawing. That you need to understand. This is trophoblast as usual. That earlier event is there. What is that hypoblast? After delamination, it was reached towards the trophectoderm also. Okay, so this green one, hypoblast, and we said this is yolk sac. So everything is there. Everything is there. But we are showing this. The total explanation is done with epiblast. How the bilaminar embryonic disc is going to become trilaminar. For that, you need to understand everything. So for theoretical aspect, now I am showing only that epiblast, the said things. Along the longitudinal axis of the embryonic disc, a primitive streak is formed. The formation of primitive streak marks the beginning of gastrulation. A longitudinal furrow known as primitive groove. Okay, the forms along the middle of the primitive streak. On the other side of it are primitive folds. I'm drawing primitive folds. Okay. Then anteriorly the primitive streak has a shallow primitive pit. Yeah, Rachel. The region in front of the primitive streak becomes that thickened. This thickened part of the streak is called primitive knot or primitive node, that are Henson's node. So talking about this one itself, primitive knot or primitive node or Henson's node. The primitive streak and Henson's node provide place and avenues for the migration and ingression of the future mesodermal and carda mesodermal cells to their respective places for further differentiation. The bigger line need to go for that. That ingression of the future mesodermal and carda mesodermal cell to respective places for further differentiation. The process of migration of cell is called gastrulation. The process of gastrulation transforms the process of the gastrulation, the transform the two layer is into three layer. I have to explain this. Two layered embryo into three layered. So that here little expansion required. So for that, I do once again some in the information in the lower paragraph and some information here. So let me not to confuse. So I am okay. Blue color. Epiblast. I'm drawing epiblast with our hypoblast is there. So how bilaminar embryonic disc is going to turn into, imagine the trophoblast is as well. Okay, so then the primitive streak, primitive knot, primitive streak, and the primitive folds as well. Now, I am using my own words here to make it. The ingression of epiblast cells, replace the hypoblast cells for the formation of the ingression of epiblast cell, replace the formation of hypoblast for the formation of replace the hypoblast for the formation of endoderm, Next, some of the future mesodermal cell, that's what they talk about here. The future mesodermal and carda mesodermal cells will enter into the primitive groove. They reach in between these two. That means epiblast and that endoderm, and they form the mesoderm. Here only I explained the three layers formation. After the formation of mesoderm, that means you need to understand epiblast only formed endoderm. Epiblast only entered the primitive groove and reaches between the epiblast and endoderm that form the mesoderm, and this is endoderm. Okay, then this is epiblast. Now you can say ectoderm. The red one in the middle, that is mesoderm. So now the trilaminar embryonic disc is formed. I'll show the video also to you. The trilaminar embryonic disc. Okay, so this is called gastrulation. Okay, so more we need to expand this in the coming classes. So trilaminar embryonic disc is formed. Okay, the trilaminar embryo, formation of primary germ layers. How it was happened, that I explained now. That ingression of future endodermal cells form the epiblast, replaces the hypoblast. See that whatever the theoretical information given here, that I shown in the form of diagram. ingression of the future endodermal cell form the epiblast. The form the epiblast replaces the hypoblast and form the endoderm. Here I shown this blue line. Okay. Then the future mesodermal cell converge towards the primitive fold, this area. That future mesodermal cell converge towards the primitive fold, move through the primitive groove and reach between the epiblast and endoderm. Reach between epiblast and endoderm. The remaining epiblast now constitute ectoderm. Thus, the three germinal layers, namely ectoderm, mesoderm, endoderm, is formed. Ectoderm, endoderm, and mesoderm, and all derived from undifferentiated cells of the epiblast. That is to be noticed. The total responsibility is belongs to epiblast. That only going to form the future baby. The future baby is going to be formed from here only. Now the doubt raises here. If we draw the full diagram, the doubt will come. This is trophoblast. We are seeing. This is trophoblast. Okay, so then after we drawn the epiblast, isn't it? We drawn the epiblast. Then after we drawn the hypoblast is there, still little bit is there. Then as I said to you that this epiblast only formed the endoderm, and in between them that by moving towards the primitive string, the cells reaches between these two and form the mesoderm. That means epiblast only formed ectoderm, epiblast only formed mesoderm, and epiblast formed endoderm. So this area we focused, isn't it? This area. What about the remaining one? So that we need to understand the complexity in case of human being. Okay, so this is also ectoderm. This is also endoderm. And one more layer is actually need to form, that is mesoderm, is also going to form here. So if this is ectoderm, mesoderm, endoderm, here also ectoderm, endoderm, mesoderm are present. What is this? That you need to understand. We have the two things here. One is the double action. Simply we say double action. That means embryonic layers, extra embryonic layers. You need to understand them as embryonic layers, extra embryonic layers. Okay, so type this, need to increase the size. Okay, so we need to go for this embryonic. This is embryonic one. These are embryonic layers, ectoderm, mesoderm, and endoderm. Okay, so then what is these extra embryonic layers? That we need to say they are extra embryonic layers. Okay, here only we need, I want, right? So I'll make it here. Okay, so let me write it if it is not possible. We said this is with, let me take another color, a black, yeah. So this is embryo, embryonic ectoderm, embryonic mesoderm, embryonic endoderm. What is this? Extra embryonic ectoderm, extra embryonic mesoderm, extra embryonic endoderm. What these things do? We know this is embryo is going to form the fetus, the digestive, respiratory, circulatory system. All the things is going to form. Okay, but what this thing is doing extra embryonic. That only form extra embryonic layers called amnion, chorion, yolk sac, allantois that we have to discuss separately. Now, allow me to go here, this area. Okay, so you just allow me to go here, that embryonic area. So I'm taking embryonic layer with your permission. So differentiation of the mesoderm and formation of xylem. For that, that allow me to go for embryo for today. Okay, so differentiation of embryo. So till the time we are seeing this particular the information. So our things, because of our typing, some disturbance is created. So let me go here. So trilaminar embryo we discussed, bilaminar embryo now became transformed into trilaminar embryonic disc. Now the formation of notochord and neural tube. So after explanation of this, I'll show the video. Just please focus here. Okay, so that induction process we have to learn over. This is epiblast. Now it is called ectoderm. Now the middle mesoderm, and this is endoderm. Now I'm showing only embryo tube. Okay, so the carda-mesodermal cells present in the epiblast of the embryo converge and involute through the Henson's node and extend forward as notochordal process. Okay, so this is the carda-mesodermal cells. They are saying they enter through this, and they are responsible for the notochordal. They enter through this, comes here, and they are responsible for the mesoderm. Notochord is derived from mesoderm, you know, I think. So they are notochordal rudiment. This is later transformed into a solid rod called notochord. As we know, in future, this is going to form vertebral column, replaced by vertebral column. Okay. So that visual I shown, which is replaced by vertebral column. The notochordal mesoderm induces that whenever this notochord is formed here, it induces overlying ectodermal cells to form the neural plate. Okay, this is called induction actually. So the very interesting thing, you focus here, very interesting thing. Okay, so let us take in human being that we know this is the skull. In the skull, that who is present in the skull? Brain is present. Okay, so brain. I am trying to show you that, not the full picture of the diagram. So skull. Skull is derived from mesoderm. Brain, nervous system, derived from ectoderm, isn't it? Derived from ectoderm. Okay, so that means in adult human body, if you see there, the outer skull is belongs to the derivative of mesoderm. Inner brain is belongs to the derivative of ectoderm, isn't it? That simply what I am saying here, in the samosa there is aloo. Okay, in the samosa there is aloo. Red color is aloo, the green is samosa. Okay, so if you don't work properly, that means here, if the samosa made with atta, aloo made with aloo. If you don't work proper, you can't make the samosa, inside the samosa, you can't put aloo. That means while cooking itself, you need to arrange these. That's what I am saying here. So please don't think otherwise, that this is the best example, I feel so. So there, if after putting aloo, then after I have to close, then if I cook, you will find aloo in the samosa, isn't it? In the same way, so what I am saying here, if the mesoderm, we said notochord is developed, isn't it? If notochord is developed and that is replaced by vertebral column and the skull is formed, that till the time nervous tissue is not formed, what happens? The skull will be empty, isn't it? So that means in our body, development of one structure is related with the development of other structure. That means if the skeletal issue is started forming here, they should inform to their friends that who is the friend of this that skull, the brain. So they need to inform that that I am ready, I am forming. You also get formed. That means that is called induction. The notochordal mesoderm induces the overlying ectodermal cells to form the neural plate. Okay, so when skeletal elements started forming in the mesoderm should motivate the ectoderm to form that nervous element, the neural element, isn't it? So neural element, that is, this is a good example of induction. That means simply what I am saying, the development of one organ stimulates the another organ to develop. Mesoderm is activated, started developing skeletal elements. So that activates the ectoderm. So that you need to understand. That is called induction process. So many are there. One example is given for us. And next, neural plate, talking about neural plate above the primitive knot. The neural plate is invaginate towards the notochord that has to go here, that notochord, and to form a neural groove. The slowly neural groove is formed, which deepens progressively to form a tube in the mean period of time that our the skeletal element notochord should be replaced by skull and vertebral column, and that should enclose that That means the blue one is nervous system. Here in this diagram, I am showing. The blue one is nervous system. Okay. So that slowly neural plate invaginate inside and develops into brain and spinal cord. Mean period, that red one, that mesoderm develops skull and vertebral column. Okay, so if they don't cooperate each other, what happens? What happens? Brain and spinal cord outside, skull and that vertebral column will be formed itself. So that means the one structure need to facilitate other structure, need to stimulate other structure here. So that's it. So that we have to stop here. We need to stop here to see whatever the event I said about the formation of the gastrula and the development induction procedure that I'll show you in the video. So the gastrulation process, how they are going. Ectodermal growth, just showing ectodermal growth. Okay, that means you need to follow this line. This is the blue one, ectodermal growth. So that means in this diagram, we see only ectodermal issues. Okay. So you can see there slowly the primitive streak is going to be developed here, the primitive streak, and the primitive streak is going on that form the primitive groove. This is the primitive groove. And along with that, we know that the ectoderm is going to form the neural plate. Neural plate slowly develops and form the brain. The brain is also formed from here, the brain. And the remaining you can see spinal cord. So that means here we discussed about ectoderm. So what is the things happening in the ectoderm? So till here. Okay. So let me say where we are exactly. Where we are exactly. So what is this? The totally whatever you are looking towards in the picture, that is that we need to know what it is exactly. So that we are seeing here like this, isn't it? That epiblast, and below that epiblast formed mesoderm is there. Below that epiblast formed endoderm is there. We are not talking about that extras. That means the trophoblast, that hypoblast, that we are not discussing about them. Only embryo. That as I said to you, embryo, extra embryo, three things are there. So this, the blue one is ectoderm. So what is happening in the blue that you need to watch over. Okay, so brain is developed, the spinal cord is developed. At the same time, inner, what is happening inside, that we need to say in the mesodermal growth. Okay. So for that, they are making the ectoderm as a transparent, and they taken the transverse section here, the transverse section. So now, let us see what is this. This is epiblast. Okay, this is epiblast formed endoderm. Then what I said, the ingression of epiblast cell reaches here, the transverse section from here. Ingression of epiblast cells reaches the sum of the future mesodermal cell reaches between the epiblast and endoderm, spread in all direction. They form the mesoderm that we discussed here about the formation of mesoderm, that you can see in the diagram. So there epiblast, from the epiblast. See that some of the epiblast cells will form the endoderm. Endoderm formation is over. Now, what next? What is need to be formed here? Mesoderm. So how does the mesoderm is developed? Now this is epiblast, this is endoderm. Then ingression of epiblast cell replace the hypoblast for the formation of endoderm. That's over. Some of the future mesodermal cell converge towards the primitive groove. Okay, so let me that spread in all direction and form the mesoderm. Okay, so that's it. So the formation of mesoderm is also shown to you. Little bit back we'll go. Yeah, this is the thing which I shown to you. So this is what I am showing always. So that what I said, this is epiblast, and epiblast ingression makes the formation of endoderm. So what I said, through the primitive groove, some of the future mesodermal cell converge and reaches here. reaches here and spread in all direction for the formation of mesoderm. We say, so that thing which we are showing in the diagram. Okay, the spread in all direction. How they spread in all directions, that mesoderm is formed. That you can see the red color is the mesoderm. Okay, now the full pack embryo. So in that embryo, the outside what is developing? What is this? This is brain. So in the at that same time in mesoderm, what is happening? That's what we said. Actually happening in mesoderm, that is the first thing happened. That means notochordal rudiment formation. So for that, we need to make this ectoderm little bit transparent to understand the issues. So we are making that transparent, and then you can see the notochord formation. The notochord is getting formed. Okay, so whenever a notochord is formed, automatically upper side we can see the neural plate. So I will let me use the two colors here. This is notochord that induces the upper ectoderm to form the neural plate. So at a time both will form. Future notochord will be replaced by skull and vertebral column, and this neural plate form the brain, and that encloses in the skull. So that's the story actually. Okay, so that what we said it as induction process. That we call it as induction process. Okay. So then the next class we continue with the embryonic